All right, we have liftoff. So welcome everybody. We're very, very excited to gather this diverse audience in the room tonight for a very, very special event where we're going to be presenting work from the ISS and from our research labs here at UCSF. And we're very excited to talk about why the space environment and the space challenges are really a forcing function for many of the problems we're trying to solve in terrestrial research in life sciences and also in healthcare. And I think you'll hear that theme come up over and over. I use a shortcut for that, which is why are we investing so much in space? Of course, we love the exploration, but we really care a lot about how it can benefit our terrestrial health and life needs. So instead of the ROI, I call it the ROE, or the return on Earth. So that's the shortcut for all of that said. There are many different examples of it tonight. And first, we're going to hear from someone who probably has the most experiential view of all of this, and that is Dr. Kate Rubins, who's a NASA astronaut. We're very excited to have her here tonight. We also have good friends and colleagues and UC Space Health researchers here tonight, Sonia Schreper, Dr. Tammy Chang, Dr. Sonia Schreper, Dr. Tammy Chang, and Dr. Charles Hsu. And we're going to be hearing from each of them. We'll have a discussion and we'll have Q&A, and then we'll roll back out into the reception, but not in a hurry, because this is a very rich mix of a brain trust here. So I'm going to start by introducing Kate, first of all, which is what she goes by. And as you can imagine, it's very hard to get a hold of her and to get her to land anywhere for very long. But we've been successful at doing that and having her come to UCSF and dedicate some time for us tonight. We're really excited about this. We've had many great exchanges. I now consider Kate a friend, and we've had lots of time to share ideas, and we're building more collaborations between Kate and UCSF, but across the UC system and many other collaborators. So to introduce Kate properly, I do need to read this, otherwise I'll wander off into some jokes. And I promised her 2 truths and a lie. I won't be telling a lie. But Kate is an astronaut, a scientist, also an innovator, and a world-renowned expert in molecular and cellular biology for use in low Earth orbit and low resource environments. And beyond low Earth orbit, she's helping push the envelope into deep space. So back to the moon and onto Mars, and you'll hear about that. And that forces us to look at untethered models, which we also need on Earth. At Stanford, she co-developed the first smallpox animal model and genomic tools for high consequence viral diseases. At MIT, she mapped the pox virus transcriptome and led mpox fieldwork in the Democratic Republic of Congo. Since becoming a NASA astronaut in 2009, she's led the development of molecular techniques for human health in space and served as deputy director for NASA Health and Human Performance. That's not enough. She has also become a major in the Army Reserve and regularly collaborates across government agencies. She's had 300 days in space on the International Space Station, including 4 spacewalks. She did the first DNA sequencing in space in collaboration with our own colleague here, Dr. Charles Hsu, and microgravity research on cardiomyocytes, organoids, and synthetic biology. And these technologies also advance diagnostics and health tools on Earth, as we've said before. So I am not going to delay this any further because there's not going to be enough time. So let's go ahead and welcome Dr. Kate Rubins.
Thank you so much. Thank you, Dr. Sawyer. It's just really a pleasure to be here, and I appreciate everything that went into putting this panel together, and I'm really excited that you can hear from other space researchers as well. So thank you very much for the invitation. And, I wanted to talk a little bit about the next frontier. Like, where are we going? And I'll give you a little history of where we've been with space and biology, but where are we going? And this is really a marker of where we are. I've said this is the second space age, and I think we are currently in the second space age. The first space age was all about government-led and proof of concept, like, can we survive up there? How do we build the space station? But now I think this age that we're living in is commercial, collaborative, and it's science-driven. So we've got private space stations that are lining up to succeed the ISS. We've got lunar missions on the manifest. And all of a sudden, biomedical research payloads are very hot. They're competing with satellites for upmass. And so why did we get here? And why does this matter? And I want to take you through a little tour of what it's like to do science in microgravity. Essentially, microgravity is this blank canvas where cells assemble in 3 dimensions. Proteins can crystallize with much more perfect atomic perfection. And biology reveals results that gravity hides. And so we'll take you on a short tour of the space station and what we're doing there. So I want you all to take a deep breath and picture yourself on the space station. This is one of our windows of the cupola that looks down at the planet. And you actually are floating heads down, which is why the Earth looks like it's up above you. So you've just floated in here heads down and you're looking out the cupola at this beautiful Earth. It's wintertime, it's locked in ice. And all around you is the framework of the International Space Station. And this is hurtling around the Earth at 17,500 miles an hour through vacuum. And up here, gravity's barely a whisper. So cells float, fluids refuse to settle, and biology behaves in ways that we just simply cannot replicate in Earth-based labs. And so this is what I want to talk about today, because when we take away that force of gravity, which is oftentimes so much signal that we're losing the smaller signals, all of a sudden we can expose new pathways for discovery. And we hope that these pathways are going to help improve our understanding of human health and disease back on Earth. But first, how did I get there, right? Well, it's a fun story, right? They load you. I launched both times with the Russians. They load you into the Soyuz capsule like cargo. This is me. My commander took the picture. So he's not in the capsule yet. He's going to sit in that seat right next to me. And then our other flight engineers next to him. And then on top of that, they pack a bunch of additional cargo. So I don't know anybody that's had kids that's ever done the egg drop experiment where you try to protect the egg and drop it from a tall tower. That's essentially what we feel like by the end of this cargo packing. The guy that's closing out is almost with his foot trying to shove us into the capsule. So the closeout crew leaves. They get far away because we're about to do something extremely dangerous and they don't want to be anywhere near it, which just leaves the 3 astronauts and cosmonauts sitting up inside this rocket. And you have about 4 or 5 hours to sit there before launch because there's a lot of quality control checks on the rocket. And so I sat there and I thought about all of the bad decisions in my life that had led me to this point of being on top of a rocket about to launch into outer space on what is essentially an intercontinental ballistic missile, where the closeout crew puts you in, closes the door, and gets as far away as possible. However, I would like to tell you everything went fine. Our rocket launch was nominal, which is the most perfect word in the English language when you're on top of a rocket and launching. And it got me safely to the International Space Station. And so this is the inside of the lab on the International Space Station. And you can see this does not resemble any lab on Earth. If you're OCD or like things to be neat and lined up, it could drive you absolutely bonkers. I spent a lot of time kind of rearranging pens on Velcro so that they would all be straight because it was the only way I could bring a little bit of order to the chaos up there. But this is the place that all this exciting work happens. And so all of these experiments that we bring up there also have to survive launch vibrations. They usually have to fit inside a shoebox. They have to sip only a few watts of power, and they've got to keep the humans safe while we're doing these experiments. And so wrestling with all of these constraints really forces us to redesign things that we think we've got scientific equipment on the ground and we don't think about it, right? We open up our Fisher VWR catalog and you order a centrifuge and things just come and they are the way that they are. But when you have to design for the constraints of a laboratory like this, it really causes you to think about equipment and about your experiments in a completely different way. And so this sparked something it's a question that I'll keep returning to today, which is how can this radical engineering that keeps people alive at 17,500 miles an hour in a closed environment in the vacuum of space, how can that transform how we live down here on Earth and how we treat human health and disease? So why microgravity? I talked about this a little bit. Essentially, this is a really unique laboratory for scientific discovery because if you can think about gravity as a 9.8 meter per second squared soundtrack that's turned up really high and is drowning out these quieter notes of physics and biology, when we turn that sound down, all of a sudden some patterns are revealed. And so these are specific things like patterns with buoyancy, convection, and sedimentation. So we hit the mute button on this loud soundtrack in space. And this lets us actually identify fluids that would normally slosh around and stratify instead of shape themselves into perfect spheres driven by surface tension and adhesion. And it also lets us take a look at what happens with cells and cellular structure and how things assemble and organoids, and in ways that we can't do when we have gravity pressing them on Earth. So this mimics potentially the in vivo architecture a little bit better. We've got proteins and materials that behave completely differently. So the proteins can crystallize with much more regularity, which really reveals atomic details that we couldn't see otherwise. And so microgravity is not just kind of this novelty. It's like a clean room for fundamental science. And so we get rid of this gravitational noise and we can kind of see all of these really interesting things that are going on with our science. And so I just want to tell you about a few case studies for biology research in space. So we'll talk about the DNA sequencing in space, mostly because it's the coolest thing ever. And Charles is here and it's still my favorite experiment of all time. And we want to talk about cell culture and organoids a little bit. And kind of what we're doing in space and where we're going to go with some of these organoids, and human health monitoring, water purification and sustainability, and synthetic biology and biomanufacturing. So let's start with the DNA sequencing in space. So why even do this? Why did we get this, bug in our bonnet about doing DNA sequencing in space? And really, it's because the way that we used to do environmental testing of microbial contamination, or if we had some infection that we were concerned about on space, is we'd have to wait until a cargo vehicle or a crew vehicle was coming back to Earth. So we got to wait until that Soyuz is ready to undock from the space station. That only happens every 3 or 4 months. So at that point, we could take a sample of what we were concerned about, green fuzzy mold growing on the wall where people are hanging their towels. That happened. We're not allowed to hang our towels there anymore. So we have environmental concerns on the space station. If you had somebody get sick and you really wanted to know what's going on, we don't have a clinical laboratory up there to diagnose this. So you would have to take that sample and you would have to wait until somebody's coming home on a vehicle, streak it on a plate, wrap it in parafilm, put it in the Easter egg, box that's hurtling back towards the Earth. And then NASA sends a plane out to Kazakhstan and picks it up, brings it back to Houston, and then we start standard microbiology processes. And this is just not a time scale that bacteria respect. And so if you have an actual infection that you're concerned about or a real microbial environmental threat, we can't do these things this way. And so we wanted to develop something that was going to be point of care, immediate readout of something that we were, had a concern about, or things that we just wanted to look at to see the microbiome of the space station. And so, you can kind of see this is me doing one of our standard routine swabs. We do swabs all over the space station to make sure that we understand the environment. And space is super fun because you get to do your lab research while you're hanging upside down from the monkey bars. So it kind of just like brings out your inner kid no matter what, even if you're swabbing all over the place. So this microbial demonstration was kind of the driver. But sequencing is a platform technology. So as we thought about this, there's a lot of other things that we could do once we have this platform working. So if you can think about, maybe we'd want to do biomanufacturing in space, and you always want to know what's going on with your bugs. Like, do they still have their payload? Are they mutating? So we could do quality control from that. We can also profile the human, what's happening physiologically. We could get readouts in real time. Like I said, we don't have a clinical lab on Earth. Right now, our medicine is based on the fact that we can get that human back to Earth within 24 hours. That's not going to be the case for the moon and Mars. And so we need to just start developing this real-time clinical capability. We want to increase this Earth independence, and we're going to be forced into this as we do moon and Mars missions. And then really long-term down the road, some of these methods could be used to detect non-canonical bases, the search for life on Mars, and things that we're doing for astrobiology. So lots of reasons to do this, and then lots of concurrent challenges. So as we set this up, this was a program that was just going to fly some hardware. We thought it was going to break in interesting ways. We were going to see what failed, and then we were going to refly it and do that again. And so I'll talk about some of these challenges that we were encountering. So the first is power, mass, volume. So you can see on the right-hand side, this is your standard benchtop sequencer. Really, the Nanopore machine solved a lot of this. And I don't have any particular allegiance to Oxford Nanopore. If another machine comes along that's better, I'm going to use that. But that's what we had. It's a pretty good little machine, and it was fit within the constraints of spaceflight. It's small. It doesn't have a lot of power. You can plug it into a tablet. It runs off of that. It's easy to launch. You're not trying to align a laser that's gotten, shaken up and vibrated on this rocket launch, and you don't have a CCD camera on it. So lots of reasons that this was a good technology. The next thing was fluids. So fluids behave really strangely in space. So you've probably all seen, the water droplet and the astronauts like doing cool things with water droplets, but that actually it's really hard. So we probably spend 20 minutes before we go out camera and film it just by like squeezing the water bag and trying to get the droplet off and like get it perfect, because it'll stick to anything and everything. These forces of adhesion are absolutely dominant. And so, you've got to be really careful with this, because a close personal friend of mine was doing a demonstration for kids and decided to do this cool demonstration. And I was going to, make this ball of liquid. And so I used green tea, and it was going to be really cool. And I was going to sip the green tea. Well, I just smashed my face into it and kind of went everywhere in my eyes and like up my nose as I'm doing a live demonstration for a bunch of children. And there's some coughing and snorting, and we got it all sorted out at the end of the day. But I've learned to like actually test drive my demos before I do them live in front of children. And green tea was not the best choice for something that might end up in your eyes. So we're going to work on the safety as well. But you can see these fluids behave so differently. Also, if they get bubbles in them, those bubbles are going to stay. They will be there forever, because you don't have the air naturally rising to the top and the liquid settling to the bottom over time. And so this can create a big problem. If you have a big bubble that's over a bunch of nanopores, your sequencing reaction is not going to work. And so some of the things that we did, some of this was like on books, and some of this was just me, a little off books, trying to get some of this stuff done, was sent up a bunch of lab equipment to space to see, could we pipette in space? How is this all going to work? And when I got to NASA, initially, I started asking about the molecular biology lab setup. And as it turns out, we didn't have one. We would build these shoebox-sized payloads that were like an experiment per payload, and they'd be like a shuttle mid-deck locker. And it would go up, you would push the button, the payload would run, you get your experiment. But there wasn't really a way for the humans to interact with this experiment, to, do all the things that you need to do in a lab. And I said, I was very early on in my career. I was an ASCAN at that point. It was just what we call astronaut candidate. So this will tell you how junior I was, that my title was ASCAN. And I started asking these questions. And it was I was just told, silly ASCAN, we don't pipette in space. It was variations of we can't pipette and we don't pipette. And it just made no sense to me. Like, why in the world do we not pipette in space? And so when I got ready to fly, I sat down, we have a psychologist and a psychiatrist per mission, and we get a really small allotment of like things that are supposed to keep us happy in space. And so you can see where this is going, right? People send cookies or brownies or like a stuffed animal. And I was like, no, there's a ton of food up there. I'm not wasting my up mass on brownies. But I really want to send lab equipment because that's going to make me happy. And so I got the psychologist on board. We had a session. I talked about it, like what brings me joy in life. And at the end of it, he said, you know what, we do need to get this on board. And so we had to send it to the safety committee, and it went through, all kinds of approvals and everything. But at the end of the day, we got this stuff up on board in my personal psychological support kit so that we could start doing laboratory experiments in space. And I'll show you just a little bit of a video of these kinds of things. And you can see there's challenges that we have, right? Like stuff is stuck to the table with duct tape. The pipettes are upside down. You have to think about where you're putting your tips. You have to hold everything. But at the end of the day, this still works. This is air displacement. This is positive displacement. We have filter tips. What's the structure of the material surrounding the liquid, as long as the surface to volume ratio is really high, it stays really nicely in the container. And so, I just did this stuff with like simple food coloring, and I filmed this. and I'm a tad snarky. So I just sent the video back down to the ground to the people that I talked to that said, we can't do pipetting in space because, I mean, you can't do that in your tissue culture hood. Like, don't try that at home. But we also had to develop some equipment for this molecular biology suite. We don't have a lot of centrifuges up there. So you get really creative. And we do have a 3D printer. And so we printed a rotor and put it on a drill. It works great as a centrifuge. And so, you know, this is, you have fun doing these kinds of things. This is really, this is what science is about. And I do think that these space constraints let us be really creative with that. So the end, you know, long and short, this is the sequencing. It worked. It worked really well. It worked the first time. Like, I'm a scientist, you know, and I've had a lab and I've done a lot of research, but my grad students and postdocs, you know, there's kind of the time in the PI's life where they start to hide your pipettes and they're like, just, you know, go back to the office and write your grants. It's fine. We got it covered in here. And so like, I was a little nervous, like, hey, is this going to work? Right. And if you've ever had a time to have an experiment work perfectly, it's when you're doing the first DNA sequencing in space and there's like 300 people watching from the ground. So I was pretty excited that this all worked. And you can see this is the publication that we did with a whole bunch of folks, and Charles was incredibly instrumental in this effort. And so we sent a mixture of E. coli and lambda and mouse genomes. We wanted a range of GC contents, and we wanted a mixture of genomes so that we could actually try to reassemble and identify species out of this complex mixture of genomes if it actually worked. But for this, we wanted to focus on the hardware. So we prepared the libraries on the ground and we sent them to the space station, and then we really tested the hardware. So after that, the hardware works. And so now we wanted to extend this to be able to go for this complete swab to sequence pipeline. So we did DNA sequencing, direct RNA, and cDNA sequencing. So then we wanted to go collect some bacteria from the wall, streak it on a plate, and then go into the sequencing reaction, actually do the library prep on board. And that worked incredibly well. And then we wanted to skip the plate and try to do culture independent profiling. We've done that of surfaces and of water. And the water is really interesting and very recent. And it's caused us some challenges because you have to filter it. But that's just really recent work that's coming out. And then there's a really cool student project that actually did CRISPR-Cas9 transformation and use sequencing for the readout. So you can see we've expanded this technology. It's a platform up on board ISS. It's available for experiments. I'd like to use it more for human health monitoring. And I think that's something that's super exciting in the future. I'm also really excited about biomanufacturing, and I think it'll be a critical component of that. So where do we go with sequencing in the future? Automated sample preparation. Like the sample preparation, the library preparation still involves a lot of pipetting. And so it's kind of funny. We did the pipetting in space and demonstrated that it works. And now I don't want to do it anymore. I want this all to be done on a microfluidics chip. But we want to really speed this process up. And we want to make sure that we could do it at higher throughput. Miniaturization and modularity. And so identifying all of these components of the sequencing reaction, getting them as small as possible, getting them modular so that you can swap and interchange some of these different things. So I think the high throughput scalability is important. And I'm not talking 1,000 samples. We don't need to sample 1,000 samples in space, but it would be really nice if we could do 96 samples, barcode them, and then run those through the sequencer all together. I think really where this is going is eventually we want to get this hardware designed into the life support system on the space station. The space station is this crazy cool place where it's a completely closed loop. So, we recycle our air, we recycle our water. This is possibly very interesting from a microbial standpoint. And so getting sensor systems that are actually embedded into these areas is going to be really interesting. And I'll talk a little bit about the water system later on. And then eventually, we want to build models of this. I'd like to do machine learning with really large scale microgravity data sets. And so one of the most recent things we did with Rob Knight's lab at UC San Diego was we mapped the entire microbiome of the space station, just spatially, not temporally. And so this was Kate hanging from the ceiling doing 1,000 swabs of the space station. It was a really long week. I got through it because I put on like my Apple playlist. I started with 60s rock anthems, and each day got a decade, and then we got 1,000 samples at the end of it. So everything was fine. We shipped those back to Earth. But that's kind of one of the things where we can see this really going super high throughput to do this spatial map. And that was published pretty recently in Cell, if you want to look that up. I don't have a citation here. So I want to switch gears from the sequencing to cells and organoids. Let's like zoom out a little bit from the molecular level. And so here, again, we didn't have long duration cell culture. We had shuttle flights that went up for maybe up to 17 days. They had their shoe boxes. The astronaut/monkey pushed the button and the treat came out the other end. And we got our data when the shuttle came back to Earth. But we really wanted to expand our ability to culture cells, long duration in orbit. And as you can imagine, there's going to be some challenges with this, right? How do we grow these cells in culture? Like I showed the 96 well plate and things don't really cross over. But if you've got cells and cell culture, you have to worry about contamination. And so, how do you change the media, right? Like on Earth, you just suck it up with the vacuum and then you add some fresh stuff. When fluids are behaving how they are, and you don't necessarily have easy access to vacuum lines, despite being surrounded by a complete vacuum, we got to reinvent just these processes for how we're doing cell culture. And so there was a lot of work done, and this effort was led out of Joseph Wu's lab at Stanford. And so what we did was we took PBMCs, we made iPSC cells, we differentiated them. This is all on the Earth, and then we split half. We left that on Earth. We sent half to space, and we grew them. We cultured them for 30 days. And then we did some analysis on board, and then we also were able to send the cells back live. They landed off the coast of Long Beach. They got picked up on a boat. They got driven up to Stanford, and then they started doing the, like, can you imagine, like, the grad student, right, that gets sent down to go get the cells from Earth and drive them up the coast of California. That I thought that part was just one of the coolest things of this experiment. And so we did these experiments, and you can see this is from one of the more recent experiments that we've done with the cell culture chamber. We also had a 6-well plate that had a gas permeable membrane, and then the bottom was an optical quality bottom so that I could look in the microscope every few days and check what was going on with these organoids. With this, we decided to suspend it between two pillars so we could use microscopy and automated video analysis to look at contractility. And then, of course, we did formalin fixation and we looked at gene expression. But it was the payload comes up and everybody looks at me and they're like, Kate, you're the biologist, you're taking care of these cells. And like, I'm okay at tissue culture, but I'm not the best. Sometimes my students would kick me out of the lab and they're like, we just really don't want you to contaminate stuff. We need to get this done. And so I was terrified for the 30 days that I was handling these cells, that I was going to contaminate this and ruin the first heart cell culture off of Earth. Luckily, again, like if it's going to work once, thank goodness, I did not contaminate these cells. It was like an unveiling, like every week when we checked the cells, like everybody would come gather around because they'd heard me stressing about it all the time at the dinner table. And like, did you break it? Did you mess with it? Is it broken? And I'm like, no, they're not contaminated. And they clap for me because they're good crewmates. So we followed up with the initial experiments, which were just cardiomyocytes. And you can see this in this paper here with those iPSC cells that were actually engineered heart tissue. So cardiomyocytes plus endothelial cells plus fibroblasts in this work. And so this got expanded into this really cool collaboration between NIH, the National Center for Translational Science, and NASA. And we've sent 10 different researchers tissue chips up there. So we're looking at organoids that model all kinds of different systems. I got to do a really cool experiment with bone and synovial fluid and looking at inflammation and treatment there. We've got liver organoids, which you're going to hear a little bit about. We've got brain organoids. And so this is a way to be starting to study some of these systems on board. This was so successful that NIH and NASA decided to fund a second version, Tissue Chips 2.0, and that's actually ongoing right now. And so we're actively flying these experiments to space station. And so if we keep zooming out even further, we've gone from DNA to cells. Now let's get to the human and talk a little bit about human health in long duration spaceflight. And I will preface all of this with, I love astronauts. I'm an astronaut, they're my friend, but I don't really care that much about astronaut health. I'm really interested in the human health back on Earth. And so the great thing about spaceflight, well, maybe not so much the great thing when you're the astronaut doing it, but it really debilitates you when you come back to Earth, and you got all of these hallmarks of aging. So you've got a lot of neurovestibular disorders. You get things like arterial stiffness. we have a fluid shift upward that really messes with your body systems and how the fluid distribution is happening. Certainly with the circulatory system, there's an interesting article in New England Journal that's a case study of an astronaut. We were doing routine ultrasound because that's kind of what we've got up there. And this was to look at arterial stiffness. They happened to notice a clot in the jugular vein. And you know, like, right, everybody panics. And how much do we have in terms of anticoagulants on board? But then if you put somebody on anticoagulants and the space station loses pressure or has a fire and they have to deorbit, they could have serious trauma upon landing. And so this was a big, you know, this was a lot of doctors talking a lot about cardiovascular, but it just goes to show you strange things happen to the body in space. We definitely have a lot of bone and muscle loss. The bone we've really ameliorated with resistive exercise, but we have to do 2.5 hours of really heavy weightlifting and running just to give us enough stimulus to keep the bone we have, or maybe not lose so much of it. We have a lot of increased CO2. This is 1,000 times higher than your terrestrial CO2 environment, which leads to things like brain fog, confusion, irritability. Not a good recipe for 6 people stuck in a tin can trying to make sure that it doesn't kill them on a daily basis. We've got really interesting immune decrements. I don't think we've studied this enough. I think there's a lot to do here in terms of looking at the immune system. We know that we have immune decrements. We see really fun things like viral reactivation. So people get shingles on board. We've got EBV levels that are going up that we can measure by PCR. Nutrition is an issue because we have to provide nutrition with shelf-stable food that's going to need to last for up to 3 years because of the way we launch things into space. And of course, we've got to collect all this waste from the humans, and we have to deal with radiation. So you kind of have these like structural problems combined with these really interesting aging problems on board. And we can, you know, I don't have time to go into all of these, but I can talk at length about any one of these and the research that we're doing. But I think it's allowing us to study things like a really healthy, normal 40-year-old that's going up into space and all of a sudden has these things that mimic aging. You come back and it's even worse because now all of a sudden you're in the full weight of 1G. And when I got back to the planet, I felt like I weighed 300 pounds and I was attached to the surface by magnets. And I got really worried that this was like a permanent disability. I was going to feel gravity all the time. Luckily, it goes away. Like you stop feeling gravity after about 30 days. But it really affects all of these systems. And you look like somebody who's aging and has a lot of the infirmities that we associate with aging. And so, but then we bounce back and we go back to being a healthy 40-year-old. And so what can we learn about these systems by mapping this? We're going to send the astronauts into space. They're going to come back anyway. This is no increased risk to us. So let's go ahead and study the bodies and see what's going on. And so I think in the future, where this is really going is much more wearable sensors. I would really like to see more human-centered devices. And so in space research, we do a lot of clinical research, but we tend to have things where it's not designed to be a consumer device. So like we wear this ActiWatch, which kind of gives you an irritation on your wrist and doesn't do anything particularly useful for the astronaut. You can't see your sleep data in real time. We've got to wait for it to go back to the lab and then computationally analyze, and then they'll tell you how you slept 2 weeks ago. And like at that point, I don't even care. And so we're working on things like, hey, why don't we bring an Oura ring up there? Why don't we have an Apple Watch that can measure sleep? In terms of the human research, you know, what do we have available? What samples do we have available where we can study all of these processes that are involved in accelerated aging? I think one really cool thing with the organoids is maybe we have digital twins of the astronaut. Like let's make iPSC cells of the astronauts and grow those along with the astronauts themselves and look at what's happening to various organs in your body. I think also we can use space as a test bed for remote biomedical devices. We have all of these constraints. We need to build this hardware to go to Mars, and it's going to provide you a platform to test things that would work really well in remote medicine. And I'll talk about this later. And then I think transparent monitoring. So anytime, so as an astronaut, you can sign up for all these IRB controlled studies. And I signed up for like 25 studies. I signed like this much consent forms. And so, all of this monitoring and being in these studies it can take a toll on you. Feel like every 5 minutes I've got to go take a cognitive test, and then I have to do my 48-hour urine collection, and then I got to go draw my own blood. And so how can we do this as transparent monitoring, where this is just baked into the system, right? Maybe it's an inline sensor in the urine and waste collection system. Maybe this is voice sentiment analysis. Like, you can see how excited I get about spaceflight, right? Like, let's just use the call center data to analyze all of the voice that comes down all the time from space and is recorded. And so I think there's a lot of potential here for doing things in the future. And by the way, if we're developing all of these transparent monitoring things for the astronauts in space, like, how nice is that for patients, right? I mean, where we can get more data from them in the comfort of their home, and it's not something that's going to be a big burden on them, I think is incredibly useful. So I want to talk about water purification just because it's so cool. This is a short one. But we have a 97% closed loop efficiency on board the space station. And so you can see this is decades of research. And if you take a look at it and it looks like it's like a totally 1980s movie, it's because it is. It was developed in the late 80s and the early 90s. But these are our racks on the space station. You can see they're kind of the size of like a really big -80. Here's a guy standing next to it. And we have 3 of these. And those take all of the condensate from the air, and they also take all of the human urine. They suck it all in, distill it down, disinfect it, and produce pure, sparkling, clean water on the other end. So the astronaut joke is like, today's coffee is tomorrow's coffee is tomorrow's coffee. And it's not just your coffee, it's your crewmate's coffee. We share everything up there. But it tastes fine. Like, it's okay. We don't mind it. This is what rehydrates our food. This is how we live. But the fact that we've done this and we're 97% closed loop efficient, we don't ship water really up from the planet anymore. The only water we ship is like technical water for use in the spacesuits. It needs to be a really specific purity. We just drink the same water over and over again. And like, imagine what this means for sustainability. So, we need to push this further, right? And so we've got these systems, but we're thinking about what we're going to do in the future. And so we've tested things like aquaporin filtration. This was a European Space Agency experiment on the ISS. Nanoparticle-based fibers for disinfectants, more like ways that we can do portable water disinfectants in a small package, non-chemical biocides, and then coatings for the water recycling system so that we don't start developing biofilms on these. And so I think a really cool part that blends potentially the sequencing with the water system is actually doing microbial monitoring. So the way that we had monitored the waste system and the water system before is we would take water samples every so often, send those back down once every 3 or 4 months, and then they would go get chemically analyzed in the lab. For the real-time inline monitoring, all we have is an organic, inorganic carbon analyzer. So we just look at total organic carbon, and that's like our rough, swag. How much is there bacteria in here? Is it causing a problem? We have no idea what bacteria it is. We haven't to date had major problems with the water system, but, bugs thrive where things are wet. And so I think this is going to be really cool data. We just started working on this last summer, so we're still getting the results back. But this is basically sampling from various parts of the water system all the way through like the black water and the gray water tanks to this end purification, and seeing like who's living there. Are there bugs there? I think there are going to be bugs there. Like, who are they? What are they doing? It's going to totally freak out the engineers. So that's going to be really fun. But it's been there for a long time and it's working. So we don't have a problem. But we really want to know what's happening with the spacecraft, because we need to build additional spacecraft to go to the moon and Mars, and we need to monitor this. And so we're thinking about other in situ sensors. But as you build this and you think about it, we're building small wastewater recycling plants that are miniaturized and modularized. Like, how amazing would this be if you had this in the hospital or in your backyard in terms of sustainability someday? And then I want to talk a little bit about synthetic biology. And so because we've had all these closed loop system and we live in a closed loop system, I think synthetic biology could be a really big player in this. And so certainly for these planetary missions that we're talking about 3 years for a Mars mission, potentially, we're going to have to be able to produce more things in situ on the way to Mars and then when we get to the planet. And we can use the moon as a test bed for this. So we've got incredible mass and resupply constraints. We really need regenerative adaptive systems. But we've got some interesting stressors that are going to cause a problem in terms of in radiation and microgravity itself. But as we're thinking about, what are we doing with synthetic biology, I started thinking about more, how do we integrate biology with the current mechanical life support systems that we have? So we have things that capture all the CO2 from the air. We've got a thermoamine system and a zeolite bed system, and then we just turn it around and we bake it off to the vacuum. We get rid of it as a waste product. But if we could take that carbon, that's a great feedstock for bacteria. We as part of our closed loop system with the water, we dry down the human brine. And actually that's what got us to 97%. Was we used to have a brine sludge in a container. And then the engineers developed a system where they spread the sludge out across a big like filter and just evaporated the water into our air system, which then capture that condensate. I installed this on board the first time that we were putting it there, and it happened to go, they wanted it to go right above our exercise equipment. And so I installed this and then immediately became the least popular crew member, because as other people are working out, as one astronaut told me, dude, I don't like my gym being in the urinal. I was like, fair enough. So we actually called no, that's not what they sound like. Some of them are fighter pilots and they sound a little bit like that, but not all of them. So we called the engineers and we need to add filters to this. And so we're learning about how do you do these regenerative life support systems, but how do you put biology into this? Well, we've got all this great nitrogen that can go be a feedstock for plants. And we do another way that we close this water loop was the Sabatier reaction. So we take a little bit of the carbon dioxide and a little bit of the hydrogen. The way we make our oxygen is by splitting water, hydrolysis. And so we've got a little bit of leftover hydrogen. And then that gives us more water, which we put back into the system, and methane, which we dump overboard. But if you've studied rocket systems, like methane is a great rocket fuel. You can use this also for fertilizer. And so I'm talking very glibly about it. Like, let's just go do all the stuff, right? This is hard. This is really hard to engineer these small modular circular bioeconomies. But I think if we start doing it, and we also need to pay attention to the economics of it, where does it make sense to go recapture these things versus where is it just going to be a lot cheaper to ship it up from Earth? So as we do this engineering, we're going to learn about how to modularize this, how to miniaturize it, how to make it Earth independent. These are all things that, like, if we want to put this system, if we want to scale out and put these kinds of systems all over the place, in homes and labs and that kind of thing, we've got to start doing this research up front. And so this is, I think, really a driver for it, because no company is going to go try to make a personal CO2 capture system that then feeds your biomanufacturing feedstock, right? We're talking carbon capture at the huge industrial level. But for space, we're talking about what we need for 6 people. And so we're going to do this anyway. We've got to send people to Mars. This research is going to happen. And so making sure we've got this element of, like, how could this help us back on Earth as we do this? And so I think the future of closed loop systems is going to be really cool. My favorite thing to say these days is every molecule counts on the way to Mars. They're all important. We love them. They're special little snowflake molecules, and we're going to grab them all and bring them into our astronaut family. But we want to focus on things like biomanufacturing and life support, pharmaceutical demand on production. And we want to build this, the environmental control and life support is the word for all of our mechanical systems. So if we build this from the ground up and we start thinking about interfacing with living systems, and we've got a cool technology test bed for the way to Mars. I like this idea of the sensors embedded in the systems. We talk a lot at NASA about in situ resource utilization. So when we get to the surface of the moon, how do we use the lunar regolith? Can we grow plants in it? Can we make habitat bricks out of that? When we get to Mars, could we use the CO2 in the Martian atmosphere? Can we pull that in and use that as a feedstock? We need to think a lot about consumables recycling and repurposing and reducing our waste footprint. So right now, about every 3 or 4 months, a cargo vehicle will dock to the space station. It brings up 7,000 pounds of cargo, and then we pack 7,000 pounds of trash into it. We release it from the space station, and it burns up in the Earth's atmosphere. Like, I am the worst carbon offender in the world. My carbon footprint, I'm never going to recover from this, right? Like, I can drive all the EVs in the world, and this is not going to fix the sins of the space program. So we need to think about what we're doing with this waste. And it's even things like the food comes up and it's in 2 packages. There's a clear wrapper just so that you don't have the food going everywhere, and then a white protective wrapper that protects it from, heat and radiation and stress so that this food doesn't degrade over time. Why don't we make this outside wrapper out of a polymer that we can break down to a monomer that we can put in our 3D bioprinter so that I can make more centrifuges? Like, right, this idea right now, it's very open loop for our trash, and we need to think about how we close that loop for our trash. I think we could, far future, start thinking about autonomous biomanufacturing systems. things like, terraforming and synthetic cells, and really like, how are you going to build a biofoundry in outer space? And so that's for the fun stuff for people who want to think on the 20-year horizon. But if we don't get started on this now, right, we're never getting to Mars. Large owners of car and rocket factories pronouncements notwithstanding. That's all I'll say about that. So, the other thing that we can think about too is regenerative medicine. And so microgravity is this kind of really cool 3D bioreactor because we lack buoyancy and we lack shear. And so maybe we can build better and bigger tissue architectures that get smushed and flattened when we're building them on Earth. We've got continuous perfusion abilities, and we get long diffusion lengths in space. And so this could lead to some really interesting things with our tissue models. For organoids, there's some enhanced organoid self-organization. We get better lumen formation. We get reduced wall shear. And so we're just starting to look at some of these things, but I think we could have a lot of really interesting results from trying to do organoids in microgravity. We've got potential complex vasculature and bioprinting. We've got really interesting things where we can do viscous hydrogel droplet fusion in microgravity. And then we've also got different ways of looking at tissue architecture and cross-linking. So I think all of these are really cool ways to explore. And so these are all the use cases from space that I wanted to talk about. But just to bring it back really quickly to resource-limited environments. You never think of space as a resource-limited environment, right? Like it seems like it has all the money in the world and it's this very fancy machine, but it is. It's hard to get things up and back. We have limited power, mass, volume. Everybody's fighting about how many watts they get from the solar arrays. And this got me thinking about what are these analogies on Earth? And so, you design things, you design an experiment, you probably, unless you're out in global public health, are not thinking ever about the fact that power is not a given. So all of this lab equipment, everything in our Fisher catalog, these research protocols, assume you've got a big hospital and you've got a big research lab and a nice bench to do it on. But we don't always have this as a given in some of these environments. And so I would like us to question our assumptions about power, clean water, data and communication. If you want to go get some cool point of care device, the first thing it does is it wants to connect to the internet. Well, if you're somewhere that doesn't have internet connectivity, your device sometimes can just be a dead brick. Equipment space, cold chain and cold stowage, and all of the logistics. And so as I've been thinking about this more, the things that we're doing in space really, to me, resemble what we're transporting around the Democratic Republic of Congo. It's got the same constraints. It's got to survive really tough environmental conditions. Once you get dropped off for a study site, they're not coming back to pick you up for 6 weeks or multiple months. All of these, the power, the mass, the volume, the data, this is a completely different space than the space station, but the constraints are very similar. So how do you do your transportation architecture? This is how we got stuff out there. We actually, at one point I had just set up a nonprofit so I could get donations from a hospital because you can do extra checked baggage on United Airlines to like fly it into the country just to get medical supplies there. And so, I'd show up at the counter in Boston and they'd be like, oh, it's that lady again with her like 10 boxes. Like, this will be fun. But it was a really cool way to get the supplies out there, but you got to think about what you're bringing very carefully. This was the biosafety level 3 that I set up in the Democratic Republic of Congo. Our thing on board that does life science glove box looks almost identical to this. It's a little more robust than this setup in a supply closet here. But as we're thinking about this, where are the other resource limited environments that this could be particularly impactful? I think of things like rural medicine. I live in Houston and like health clinics in West Texas, anywhere a patient's driving multiple hours. If you get this equipment down to be small and ruggedized and you're pushing it out to the community health clinics, then maybe they get more of their lab work and the research done way out in the field. Clinical trials, you're doing clinical trials globally and you want to do more access and more patient sampling rather than trying to ship samples back and forth with a cold chain. You've got things that you can do point of care. Humanitarian disasters where your hospital or your lab infrastructure is destroyed. For the military, forward deployed units, trying to take care of soldiers when they are really far forward and they don't have logistics and supply lines. And research and medical care in the developing world. And I think all of these have a particular set of constraints, but they share a lot with how we develop hardware for space. And so I want to take us kind of, zooming way back out, we're way past human size now. And you can see this is an actual NASA spacecraft, and the moon is bigger than the Earth. So we are farther from the Earth than the moon is. And, for once with NASA, this isn't a PowerPoint diagram. Like, we actually built this spacecraft and we sent it. We're launching some humans. One of my crewmates on my last expedition, Victor Glover, some incredible people are launching, and they're going to go around the moon next year. And so this is really happening, and there's a huge amount of momentum for the moon and Mars. But I want to make sure that we, as we're doing this science fiction stuff, and we're turning science fiction into reality, we actually don't lose sight of what we're doing for humans back on Earth, because, this is cool and it's crazy, and see how much PowerPoint there is on this slide. But these environments replicate some places that we have some real problems back on Earth as well. And so with that, I'd like to wrap up. There are so many people to thank for the sequencing. Please pay attention to this right here, because it says UCSF on it. But this is, just the work of so many people that went into this. And so with that, I don't know if we're going to move into questions now, or we want to go right into the panel. I'll leave that up. So anyway, thank you so much for being here.
Kate, thank you. That was absolutely incredible. And I think you get the sense, first of all, that she's very, very modest, but superhuman. And also that, well, we're going to do a couple of questions. She hates this stuff. But also that the model that we often think about when you're thinking of space research is we want to use an Earth analog to get as much as we can done. But the truth is, space is an amazing analog for Earth. And I think Kate captured that really beautifully for us. So I'm going to call on our UC Space Health interns. We're fortunate to have Evan Chow with us and James Rohde, and we're going to ask you to run the mics around. We're going to take probably 2 to 3 questions now with Kate, and then we're going to move right to the panel, to the highlight speakers, and we'll have more questions later. Thanks, guys. All right. Who wants to go first? This is going to be really hard because there are only 2 or 3 questions. I saw that hand go up first.
Hi, fantastic talk. Regarding the physiology slide that you had, how long does it take actually for those symptoms to appear? And has it been confirmed with actually blood samples, or is it just anecdotal evidence at this point?
Yeah, great question. I actually took that slide out. I wish I left it in with like the time course of all these things. So when you launch off the Earth, like, you're a super healthy, like, competent person that feels fine normally, physiologically. The second you get into space, you are messed up. It feels like you've been kicked down the stairs. Like the neurovestibular kicks in hard right away. The fluid shift starts. You start diuresing like crazy. And so you get your homeostasis is really disrupted for about the first week. These things start to resolve. Like, actually, thankfully, the neurovestibular goes away like 3 or 4 days. And they start to resolve. There's a few things that are on a different time axis, like the bone density. That doesn't happen right away. You start shedding a lot of calcium, and you can get a lot of osteoclastic activity if you don't get your stimulation in really quickly. But that just kind of keeps going. The immune system degradation, we don't have a good time course on that because we haven't done those studies. What we have done are things like, 48-hour urine collection. We've got a lot of chemistries. We freeze those samples and bring those back to Earth. We just don't have a lot of point of care in situ analyses on board. So we can bring back fresher frozen blood. The fresh blood we've done a lot of the immune studies on, but like, you draw the blood and it comes back to Earth 24 hours or 48 hours later, like, those immune cells they're not doing what they were doing up in space. And so I think we can push the analytics a lot further and learn about the true time courses of this. But it is usually laboratory confirmed. And then, symptom confirmed. Is a lot of astronauts complaining to their flight surgeons about all the different things that are going on in their physiology. And so, we've got a huge database of what happens in aerospace medicine with the humans as we put them in this weird environment. Great question.
To be fair, we'll go this side.
Yeah, so I think it's really cool that you were able to do a lot of like cell cultures in the space. So I was just wondering, like, how can you really translate some of the basic like research you have done in the space to maybe like translational field, like for example, maybe inform treatment in the future on Earth?
Yeah. So I think that organoids are an example where we can start understanding emergent properties of these systems. If you don't have to do something in space, don't do it in space. It's hard and expensive. But with the lack of microgravity, we can potentially grow better organoids and we can learn more about disease. So part of it is like the fundamental research. But then there's also, how could you actually produce therapies for humans? And so Keytruda, made by Merck, is an example where they had an IV formulation. They grew protein crystals on orbit and actually made much smaller, more regular crystals. That helped them with the manufacturing process that allowed them to make an oral formulation of this drug. So the patients aren't driving in to get an infusion. We can do things like potentially produce cell treatments in space. We know that some really strange things happen with stem cells. They proliferate a lot more rapidly. They actually kind of like the engines turn on, right? And then they burn out. And so we're hoping to uncover some interesting physiology there. But in the meantime, like they turn on, we can make a lot more of them. We've got the potential to study a lot of things with iPSC cells, with immune cells. And so I think it's kind of a combination of what can we learn from the basic biology? And then what are things that we actually maybe want to produce in space? And those use cases right now are very narrow. Like it has to be so expensive that you can absorb the launch and landing costs, or something that you just cannot do on Earth. So people are working on like growing really perfect corneas in space. And so things like that are very lightweight, very, very expensive or impossible to do are going to be the first use cases. But there's a lot of interest in like in space manufacturing. And as this grows, I think that we'll find a lot more use cases. I think we got to do the basic research right now to understand like, where does it make sense to do things in space? Whereas, if we can do it on the ground, do it on Earth, right? Space is hard. Great question.
Yeah, thank you.
Fantastic, Kate. Thank you. One more round of applause. All right, we're going to move into the highlight talks. And these are going to be from our own researchers from UCSF and also UCSF researcher who's also now a Cedars-Sinai researcher. So we're going to start with Dr. Charles Hsu. And he's Dr. Charles Hsu, as you can see by his title. And he's a professor in laboratory medicine at UCSF. So he has a real applied clinical role here. He's in the Division of Infectious Disease. He's co-director of our laboratory. Many of the tests that I run on my patients provided to me very readily and accurately by Dr. Hsu. Co-founder of a really exciting company. I hope you'll mention it. It's a really exciting platform that's taken off and spun out of UCSF called Delve Bio. And he's a UC space health researcher. So I'd like to introduce Dr. Charles Hsu.
Thank you, Einar. So I'll be talking today about a project that we're currently working on. It's titled Off-Earth Metagenomic Sequencing for Diagnosis of Infections. This is a project that we secured funding for in collaboration with Chris Mason's lab at Cornell. And so for this particular experiment, we're proposing to demonstrate the use of metagenomic next-generation sequencing, abbreviated MNGS, of astronaut self-collected samples to diagnose infections in space. And the general idea is, as Kate alluded to, is that we want to deploy a portable MNGS diagnostic test that has been validated on Earth for diagnosis of infections aboard the International Space Station and have astronauts self-collect nasal swab and blood samples, process the samples, and perform real-time sequencing analysis for detecting potential pathogens, including herpes viruses. In one statistic is roughly about 30% of astronauts who are in space, they do get reactivation of EBV infection that can be detected in saliva. And so we certainly don't expect astronauts to be sick in space or to develop an illness, but we can perhaps monitor EBV reactivation as a way to identify pathogens. And simultaneously, we'll be collecting these samples and aliquoting them and returning them to Earth for analysis in a parallel fashion using our gold standard clinically validated assay. And our vision for this is that this particular test capability would provide an ability for astronauts both now and in the future, to be able to do diagnostic testing themselves untethered to Earth. And this is going to be a key resource for astronauts who are venturing to the Moon, the Mars, other deep space environments where you cannot really rely on having kind of Earth-based capabilities. And so the big problem is on Earth, we're really unable to diagnose roughly about 50% of severe life-threatening illnesses. And this includes in the hospital cases of pneumonia, sepsis, and infections of the central nervous system. On average, for these patients, they're admitted to UCSF and other tertiary care hospitals. 20 to 30 diagnostic tests for infections are usually ordered. And unfortunately, the delays in diagnosis result in high morbidity or mortality excess costs. And in addition to that, in space, as Kate said, we don't have a clinical lab. And so I feel rather strongly that we need to have the ability to implement essentially a lab in a suitcase, to be able to essentially develop a very broad target agnostic diagnostic test capability using infections as an example. In general, though, you can imagine kind of expanding this capability. If you have a sequencing-based test, for instance, you can not only look for infections, but you'd also be able to assay the host response, to assay particular pathways or genes that are being overexpressed, and be able to explore non-infectious conditions as well as infections. As an example, the two astronauts who were recently stranded aboard the International Space Station, it highlights really the challenges of returning astronauts to Earth for medical attention, and certainly as astronauts venture out to further reaches of deep space, that there is not going to be the ability to return them to Earth, say, within 24 hours. And then finally, as space travel becomes more accessible for people worldwide, a larger proportion of astronauts will have chronic medical conditions. They're not going to be as healthy as Kate is or other astronauts that typically go into space. And so these individuals may have occult infections. We also will likely encounter in the future community-level transmission of infectious diseases. And that's especially as we're seeing the advent, for instance, of space tourism, where we're going to kind of get a regular influx of individuals who are venturing out into space. And so our solution here is to really equip astronauts with the tools to diagnose infections and perhaps to extend that to really any acute illness in space. And so there's significant impact for doing this. One is that this will allow us to be, the experience that we learn will be directly applicable to our testing in low resource or remote settings on Earth. And also, it's a way to address emerging viral pandemics. We know that viral pandemics don't really come from the developed world. I mean, they really come from areas, pandemic hotspots in the developing world. And so being able to provide decentralized, local-based testing, I think is going to be critical. And you might imagine that the International Space Station is probably the most low resource or remote setting that you can imagine for doing this type of work. In addition, for sake of astrobiology, that we can use sequencing to search for life, potentially diagnose our non-infectious conditions, and then the ability to sort of provide more rapid and accurate diagnosis of infections. That's a general problem that we face here on Earth. And so the experience that we get in space would be directly applicable to our experience on Earth and the ability to do decentralized testing for infections. Finally, clinical MNGS testing is still novel. Although I've been working in this area for roughly 10 years, we still do not have an FDA-approved test based on metagenomics. And so one idea is that this will serve as a blueprint for a future lab in a suitcase sequencing-based test tool. Kate already went over this, but we have a lot of experience with prior research, so I think we're really well positioned to do this study. We have experience with nanopore sequencing on the ISS. Kate Rubins was the first individual to do sequencing in space and microgravity. We have a lot of experience with clinical sample collection on the ISS as well, including a variety of different sample types that have been successfully collected and brought back to Earth. And we also have experience with processing these samples also in microgravity. There's a lot of data now on both metagenomic and metatranscriptomic analysis of spaceflight collected samples. So we have a very rich database so that we can compare against. We're not going into this completely new. There's already a lot of data involving metagenomic analysis of spaceflight collected samples. And then finally, with this idea of miniaturization, the goal here is to develop automated pipelines that will enable astronauts to actually perform the analyses locally. And this is critically important. I distinctly remember when Kate initially produced the data, it took it took longer for us to actually receive the data than it took to actually do the sequencing experiment. To give you an idea, I think it was going at a speed equivalent to a 56K baud modem or something. That was the speed that we initially were receiving the data. So it gives us some idea still that we still face challenges with regards to transferring data across space. And so we actually were the project that was selected for the 2020 for Humans in Space Challenge. This was a global challenge that was calling for essentially pioneering startups and researchers that will allow kind of critical healthcare challenges to be addressed in space and Earth. And we had joined this challenge, and we had actually been awarded orbital launch funding, meaning that this is a company, Bar Young, which has actually provided funding for us to do this study. And so this study is going to be what we call an in-flight NGS-based diagnostic project involving various companies as well as UCSF and Cornell. But the general idea is we want to be able to essentially collect samples in space, run it on the nanopore sequencing, put it through an automated pipeline, and essentially be able to provide real-time sequence data and information, and generating essentially a final clinical report. And so the general idea is that we would provide a capability of actually diagnosing infections from a variety of different clinical samples. And we're currently working on this project. We're hoping to launch in early next year for this. And to give you some idea of the applications, we've actually applied similar assays or the same assay in various places around the world. So one example is Bamako, Mali in Africa, where we're able to diagnose cases of rabies infection, as well as dengue in Africa. I'm working with a collaborator at Stanford, Dr. Julie Parsonet, who is the assigned physician at McMurdo Station in Antarctica. And so she wanted to do this kind of sequencing. So she basically spent 3 days in my lab to kind of relearn how to use a pipette. And so she's a PI. And so when she went there, she brought the nanopore sequencer, she brought all of the equipment, she brought the centrifuge, and essentially there's no laboratory there. So she was actually doing sequencing in the clinic. And she gave me an excited call. And this was, I believe in 2019, October of 2019, saying that she had discovered a coronavirus, basically, and it's causing an infection. But if you recall the date, it was before the coronavirus. So it happened to be a seasonal coronavirus. But to give you some idea of the potential of this, even now, we actually do not have a clinically validated test that can diagnose seasonal coronaviruses in our lab. Locally, we still send it out to a reference lab to get this diagnosis. And then another example, which I'll show you an example of, is doing sequencing for emerging infections. So this is one of my research scientists, Asmita, who is doing a sequencing for identifying cases of Zika virus infection. Okay, so I want to end by talking about just two brief things, some technology advances that we're planning to use for this project that I hope will have applications for point of care testing here. So one is the general concept of looking at indirect ways of diagnosing infection. Now, currently, we're very limited. We have a lot of tools for diagnosing infections by direct detection, being able to identify proteins, being able to identify antigens, using PCR, using sequencing to diagnose infections, looking for the pathogen. We are really limited with respect to the host. They're either highly targeted, such as serology testing for antibodies, or they're based on single biomarkers such as procalcitonin for distinguishing bacterial versus viral infections. And so the general concept right now is we can actually employ RNA sequencing, and using the same assay that we're using to identify RNA viruses, the idea is now we have data from roughly 5,000 samples. So we've essentially used artificial intelligence and machine learning to develop models that can help us be able to determine what type of infection someone has, or whether someone even has an infection at all. So I'm giving you some examples here. We can actually plot. This is a linear discriminant analysis plot, which is plotting in n-dimensional space all of the samples that we have from well-characterized infectious and non-infectious conditions. And so in the future, what I envision is that we could run a single sequencing test and you'll have an unknown sample, a patient who has an acute, in this case, a patient with an acute meningitis, shown there by the black square. And you can see it's sort of like in the bacterial, kind of in the bacterial cluster, or maybe atypical bacterial cluster. And if you hone in more closely, you can see that it's very close to the cluster correspond to Mycobacterium tuberculosis. And in fact, this was actually a documented case of MTB, Mycobacterium tuberculosis. So it's a way that we could diagnose an infection broadly or diagnose an acute illness. But it also applies for non-infectious conditions. This is a patient who presented with a meningitis and was thought to have an infection. But you can see here by looking at the host response, again, by RNA sequencing, it predicts that it's a non-infectious condition. And if we look in more closely, you can see it's closest to samples that gave it a certain probability score of having been brain cancer. This was actually a glioblastoma. And it was actually this test that made the diagnosis, led to the final confirmatory diagnosis. And then finally, it actually applies for emerging pathogens. So this gives you an example where we took the same models, and models themselves were developed off of very common infections. But the general idea is like, if you bring it to the developing world, will it actually be able to identify unusual infections? So we're working with a group in Thailand, and in Thailand, they have some very interesting and unusual infections, such as severe fever with thrombocytopenia syndrome virus, which is the Bunya virus, Rickettsia typhi, Orientia, Susamaguchi. I mean, these are essentially scrub typhus. So they have very unusual infections. And you can see here that the host response, again, can help predict the kind of infection someone has, even though we never used any of these infections to actually train the models. So it gives you some idea that we could potentially use this to identify, say, a novel outbreak pathogen in the future. And I want to end by talking about two methods. One is that we're interested in developing these no extraction methods where we completely bypass the extraction step. So the idea is you can simply collect the sample, lyse it, and then immediately put it into the sequencing pipeline, basically generate the library and sequence it. And we've been able to show that you can achieve essentially equivalent sensitivity for detection of different viral infections. And so this is the technique that we're planning to apply for our current project. And then finally, I want to give you a very short video that will show you an example of what I envision this will be in the future. And so we're hoping to actually install the software, but the general idea is what you can do is you can watch the sequencing in real time. So this is actually real time it's generating. So we're able to detect in this particular sample HIV, and then within about 15 minutes, you can actually identify the cause of the infection, which is Zika virus in this case. So notably, this is not using any primers. It's not targeted. It's entirely metagenomic sequencing from a plasma sample. And then I finally have some tips for those of you who are interested in spaceflight research. One thing is, I feel that careful planning is really key for this because the problem, well, maybe the big challenge I say of doing spaceflight research is you pretty much have one shot at it. And that's basically it. So it has to work, essentially. If it doesn't work, you're out of luck. The second is automation. And that's because you really want to minimize the manual steps because every single step, every single increase in complexity increases kind of the potential for failure. The other thing is adapting to low resource settings. So there's a big push for instance, lyophilized reagents, so you don't have to maintain a cold chain, very simplified protocols, so that one thing to remember is that usually astronauts, they may not necessarily have any scientific background, so certainly they wouldn't be like Kate and be experienced with using pipettes. So it's something that you have to consider. And then certainly for terrestrial applications, I think is a really important aspect of it because you really want your research to not only be useful in space, but also be useful on Earth. The last thing would be to leverage kind of interdisciplinary multi-institutional expertise. The fact that this is really going to be, every single study that you do is never going to be in isolation. I mean, you may realize as a PI typically, or as a person working in a lab, you may be focused on laboratory work. But one thing to realize is that any study in space, and I think that my colleagues can attest to that, it really involves a number of different individuals at different institutions. And then finally, especially for long deep space missions, try to develop experiments that can minimize any tethering requirements or requirements to Earth for communication, for instance. So if you have an experiment that you could train astronauts to run in reliably without requiring input from Earth, I mean, it would be more useful for long distance missions in the future. And I'd just like to thank all of the members of my lab who are involved in the work, our collaborations with NASA, especially with Kate Rubins, with the UCSF Clinical Microbiology Lab, and also we have received funding from the US CDC, NIH, Delve Bio, and the California Initiative to Advance Precision Medicine. Thanks.
Charles, thank you. We're going to have a couple of questions after the panel, so there'll be more, but there's also a reception. So there'll be lots of people coming up with questions, I'm sure. Thank you very much for that. We're going to shift gears now to one of my other colleagues at UCSF, Dr. Tammy Chang, and we're very excited that she actually made it because she's on call tonight, and she is a liver surgeon, and she would normally be doing a liver transplant right now, but that's partly what drives the work she does. Now, someone's covering for her. Don't want you to worry, but she'll have to pay them back, of course, in the schedule. But we're really, really happy that Tammy prioritized this, and this is Dr. Chang, as I've mentioned before. And Tammy is a professor in surgery at UCSF, a GI surgeon in particular, and a surgeon scientist, and a liver tissue engineer and a UC space health researcher. And you're going to see a very similar pattern across all of these distinguished speakers, and that of being an intersection person. So maybe started in one path, but developed other specialties. And pay attention, those of you in training, about the idea of becoming an intersection person, the clinician scientist, or as we say, also the clinician innovator or scientist innovator, et cetera. So these are excellent role models for you. Tammy, thank you so much.
Well, Eleanor, thank you so much for organizing this fantastic event, and thank you for the opportunity to participate. So when I think about the work, when I think about what motivates my work, I think about this graph. And this is the trends in transplants in the United States in the past 30 years. What we have here is a line of transplants that are being done. This is for all vital organs that are being transplanted. And there is a steady increase in the number of transplants we do each year. But if you look at this sort of yellow line here, this is the waitlist additions. So there's the number of people who are added to the waiting list each year also is increasing, and it's paralleling the number of transplants at a higher level. So the result of this is that at the end of the year, there is an increasing number of people who are on the waiting list, still waiting for a transplant. And we've been steadily at about 100,000 patients, more than 100,000 patients each year waiting for transplants. And the result is that there's about a few thousand people who die on the waiting list each year while waiting for a transplant. And while this may be representing the fact that we just don't have enough organs to transplant, that's why people are waiting and not getting transplants, this is actually an underestimation of the need, because many people who could benefit a transplant are actually never listed. So when I think of this graph and I look at this graph, I think having more transplants and having more advocacy and organ donation will help, but it's not going to be the solution to match the number who need organ replacement and function for treating liver organ failure. For me, the solution is a different one, and it's regenerative medicine. And I focus on the liver because I think the need for a solution for replacing liver function is perhaps the most dire. Because of our medical and technological advances, we actually have machines that can support, at least for a little while, several vital organ functions. One of them is called the left ventricular assist device that acts like a pump and can replace the function of a heart for a little while. And this technology has gotten so good that it's actually destination therapy for some patients and is used as a bridge to transplant. For the lung, there's something called the extracorporeal membrane oxygenation, or ECMO, that was very important this past pandemic for supporting lung function for a while as people's lungs recover. And of course, for the kidney, we have dialysis that can support kidney function and support life for years for patients with kidney failure. But what do we have for the liver? Currently, we don't have anything. If someone's liver fails, stops working, that person basically needs a transplant. really nothing we have to even tie over that person, even for a little while. And why is that? Well, one of the ways to think about it is that, well, the heart is an incredible organ, but it has one main function, which is to be a pump, right? And so the LVAD is a pump. The lung, main function, gas exchange. And the ECMO is an oxygenation gas exchange membrane. And engineers, biomedical engineers figured out how to do that. And the kidney is a filtration device. And that's what dialysis is. So what does the liver do? Why haven't we figured out how to replace liver function? Well, liver has more than 500 vital functions. And all of these are biochemical reactions. We're regulating whole body energy utilization, metabolizing drugs and toxins, clears waste, synthesizes essential proteins for the body. These are biochemical reactions that are interregulated and cross-controlled. So how can we devise a machine that can do this? So I would argue that to build something that replaces liver function, we need the cell. The cell has to be the essential workhorse and perhaps the most amazing machine yet. And while the need to replace liver function is the most dire, I believe that creating regenerative therapies for the liver is also the most promising. And that's because the liver has an innate ability to regenerate. And somehow the ancient Greeks knew about this, the myth of Prometheus. So Prometheus was punished by Zeus for giving humans the gift of fire. And Zeus tied him up onto a mountain. And every night, an eagle would eat Prometheus's liver. And the next morning, the liver would grow back. And this is a particular retention of that story by Thomas Cole. And it's actually at the De Young, if people want to go see this. So the liver regenerates. So as surgeons, we know in a healthy person, we can resect up to 2/3 of that person's liver. And the liver will grow back to the exact size it needs to be to support the metabolic function of that person. In certain situations, there's also liver resident stem cells that participate in that regenerative process. But thinking broadly of scale of developing regenerative therapies for people that need them, we think about pluripotent stem cells, and particularly induced pluripotent stem cells that Kate alluded to. So these are cells that are adult stem cells that can be reprogrammed to have stem cell qualities that can then be differentiated to any differentiated or mature cell type in the body, including liver cells, hepatocytes. And of all of these, and for the liver, for all of these cell types, we can make organoids from them. So these are clusters of cells, three-dimensional clusters of cells that have greater function than if you compare them, if you grow them conventionally on a plate. And they have more in vivo-like architectural qualities. So does it matter how we make the organoids? So the conventional way, or the prevailing way that many labs or many industries now are making organoids, is culturing them on something called matrigel, which is a matrix produced by a mouse sarcoma cell line. And the matrigel can support three-dimensional structure of cells for a time, but the structure doesn't last. And this is a paper that we published a couple of years ago. However, if you put these progenitor cells developed from iPS cells in simulated microgravity, these cells will self-aggregate and develop into organoids without the need of matrigel. And the three-dimensional structure of these aggregates or these organoids will persist through time. and function follows form. And so these organoids that are developed and self-assembled in microgravity also have better function than the organoids that are developed on matrigel. So this is a heat map of gene expression, and progenitor cells are just the beginning cells of before we differentiate these cells toward liver cells, toward hepatocytes. And you can differentiate these cells through as monolayers, sort of as a sheet on a plate. You can make organoids on matrigel, or you can make organoids in microgravity. And you can see that this is more than 250 genes of what hepatocytes do, functional genes. And they're highly upregulated in microgravity organoids compared to the differentiating them either as monolayers or on these matrigel organoids. And so our thought and our hypothesis is that microgravity can be a key tool for developing organoids with high function that can be used for cell therapy. These organoids can be building blocks and that can be transplanted as cells or organoids into patients who need them. However, what our hypothesis is also that in sustained microgravity up on low Earth orbit, that these organoids can then potentially organize even further and become larger vascularized structures. So this is the hypothesis that we're testing, and we're supported by NSF and the National Labs International Space Station to test this hypothesis of if we bring these organoids onto the ISS, if we could develop these vascularized structures. And we have developed some custom automated perfusion bioreactors to test this hypothesis. And this setup will hopefully be going to the ISS soon. So I hope I conveyed to you that novel tissue-based approaches to replace liver function is a critical unmet medical need, and that microgravity may be a key tool for making organoids differentiate from stem cells and potentially larger vascular structures that can be used to treat patients. So thank you. I'd like to thank my lab, our collaborators, and our funding. And thanks everybody.
Thank you, Tammy. That's so impressive. And Tammy, Sonia, actually each of us have been influenced by a very, very important person at UCSF who's passed away just a couple of years ago, Millie Hughes Fulford. And so some of us call ourselves the Millies because we were so influenced by her. She was really the first science specialist in space on the shuttle mission. So she was an astronaut, but also a scientist, and she stayed on at UCSF and did her research for many years. And this is part of her legacy. All right. And now we have another exciting talk, and this is by Dr. Sonia Schrepfer, another doctor. Feeling a little like I didn't try hard enough, just a doctor. And Sonia has done some very excited work to discover and really reveal the nature of immunosenescence and the effects on microgravity. She's a professor at Cedars-Sinai now, but she's still a UC space health researcher and colleague of ours, very close. She's in the Department of Surgery and Regenerative Medicine, and she's a scientific and founder of a very exciting company called Sana Biotechnology. You should look that up. Here's someone who is really truly an overachiever. During the pandemic, while all of us were doing something at home, she was taking a company IPO in the middle of the pandemic. And she's also done quite a bit of work on Earth, but she has done missions in space with her work. So we're going to hear about that as well. Thank you.
Thank you, Ino, and really great to be here today. Really, thank you that you invited me for this amazing event. So I want to show you a little bit today an overview of what we are doing, because our journey started in 2013 with the first NASA grant. And our interest of the lab is really understanding how space conditions affect immune aging and also stem cell aging, because the immune cells, when they age, they're really everywhere in your body. So they have contact with all other cell types in your body. So if they are now senescent and they don't function anymore, how does that now affect your body own a stem cell niche to repair? For example, if you have a vascular injury in space, or if you're an aged patient, or what happens with wound healing, etc. So we studied that with many different cell types, and how we approach it is we need from healthy volunteers immune cells we can send to space, but we would then also need the stem cells of the certain organs. So we studied the liver, we studied the skin for wound healing, we studied the vasculature, and I cannot go to a healthy volunteer and say, could I have your blood and can I punch your liver? So what we do is we use induced pluripotent stem cells. We have a blood sample from the patient or from the volunteer where we have the immune cells, and then we reprogram them to induce pluripotent stem cells, and then we program them into what we are interested in, like endothelial progenitor cells, mesenchymal stromal cells, liver progenitor cells. So that is basically the overall aim from our studies. We did start in vivo on the left side, really 2013, when we got the first NASA grant for rodent research. And we were part of rodent research 1 and 3, where really mice came back and the frozen car cases were dissected, and you had scientific teams here at NASA Ames sitting around a long table, and every team had a specific interest. Back then we had a really huge interest in vasculature. So we wanted to study in space, because of the fluid shift, why the artery, the carotid artery, is thickening and stiffening, because we see that in our patients when they're aging. It's a huge issue. And it happens actually in microgravity, this protection mechanism, that the carotid artery becomes thicker to prevent that too much fluid goes to the brain. So the first studies we had funded was really investigating the carotid arteries, and how that affects the astronauts, but even more the patients here on Earth. And then later, we were really lucky to, were funded by the NIH cases. So we were one of the teams for the CHIPS in Space program, had a wonderful NIH partner, Lucy Lowe. She guided us through a lot. So we loved that mission. Then at that mission, we shifted to the immune cells and more to the tissue chips, because mice in space are great, but mice cells have different receptors. You really want to learn about your patients. So the next step was those tissue chip program, where you can now send human little organoids or immune cells, and we actually sent the immune cells and the stem cells together, that they could communicate in those little tissue chips on space and study what happens. And when I talk about aging, I don't mean necessarily with immune aging that the patient has to be old. We have patients 30 years old where the immune cells are just exhausted. They're senescent. That can happen when you have a permanent virus infection, for example, CMV, harmless, but stays in your body. And that can permanently stimulate your immune system and exhaust it. So when I talk about aging, I mean immune system that is senescent, meaning it cannot really protect you anymore against bacterial infections, viral infections. So it's really a senescent state. And I want to show you this video here. And let's see if I can make that work. So those are the mice actually from rodent research 9 in space. Let me see. And they love the microgravity. Let's see if I can. No, I cannot really hear. Yeah, awesome. Yeah, so you see how they are floating, then do their little saltos later. So they really adapted quite well to microgravity, and we had the Black 6 mice, and they, so that rodent research mission that came back actually alive. So we went to Loma Linda, I think it was back then, and did the live dissections with a whole team. Again, everyone got another organ. It's very efficient. As a scientist, you will love it because you have one focus, and then you have next to you someone who's interested in the lungs and then the brain. It's just amazing how awesome that is to work together. And what we found, that paper is hopefully published soon. Those mice were on the space station for 40 days. That really, if you look at the bottom, the cross section that the arterial wall, the carotid wall was really thickening in those mice. And then we asked Scott Smith and Philip Albay for some astronaut data. So we had then the correspondence ultrasound from 8 astronauts. And you can see also the yellow and the red lines that what occurred for the mice occurred for the patients. I don't go into much detail, but really the wall gets stiffer and thicker. And then, of course, we are scientists. We want to understand what it is. So we spent a lot of time, I think 3 years at NASA Ames, together with Eduardo to understand what it is. And we found the mechanism. And we could actually stop that mechanism and prevent that from happening, which is amazing for us on Earth, because if you don't want a stiffened carotid artery, you need the blood to the brain. But then we discussed in the team, you really don't want to have that in space. So it was nothing we could go back to NASA and propose, oh, we can prevent that in astronauts from happening, because it is a protection mechanism by the end of the day for the astronauts, that not too much blood goes to the brain. So basically that research unfortunately stopped then there, and we shifted to other questions because it's really not helpful for the astronauts. But we learned a lot for Earth, and we want to go and study that mechanism, but for our patients on Earth and not for the astronauts anymore. Then for the tissue chips program, this is how a tissue chip looks like. They're kind of this size. And we had a lot of those, as you can see, in the incubator. And we plated the immune cells, the stem cells, and both together, that they would communicate. And they were sent to space. And you see here in front of the little incubator, Alex Gerst was the one who, I think, did in 2018 those experiments. And there was not much hands-on work for us to do, but putting the samples into the incubator. And those chips were either frozen on the ISS, and they returned frozen, that we could look at the immune cells and the stem cells, what happened in space. So we did RNA sequencing. And then some of the chips returned live. And we had actually 3 missions on that. So everything was a little bit staggered. And what we learned, I just want to give you the high level, is, for example, that the immune cells, the PBMCs we had sent from 3 healthy volunteers, you see that the gene pattern changes in terms of G is for ground and F is flight. So you see how those different genes are upregulated in flight. And if you look at the left, they're all associated with either a senescent immune system with persistent infections or tumor or aged patients. So we could actually really prove the hypothesis that the immune system is aging in space. We think it happens probably quite fast, within a few days, but we couldn't do a time study yet. We hope that we can do that in the future. So we understood the immune cells are actually aging. The question was now, what happens to the chips when we have them in co-culture with the stem cells? I show you here an example for the stem cells, the mesenchymal stem cells. They are important, for example, for wound healing. When you have a wound, they have to cover the wound and heal it. And what you see is the MSCs alone, the stem cells alone, how they increase the characteristic that is important for them to heal wounds, so that stem cell character gets really better in space. And you see in the middle, when we add that now with PBMCs and they age, how they affect the stem cell niche, and there is no wound healing anymore. So that was the RNA-seq data from the space station, but the chips that returned, we were able to do some monitoring over time. And the idea here was like, oh, we get them back and let's just reverse aging. That will happen. So that was actually the hypothesis of the grant. But unfortunately, that was not true for all samples. It was actually true for the stem cells we sent alone. So you see here in green, those stem cells, the mesenchymal stroma cells, you have them in a dish and you make a scratch. And now they are supposed to migrate into that scratch. And you see that in the baseline, in the ground control, how fast that should occur. So they are really covering that wound. It's a wound healing assay. So then here, the 4 videos, those are the chips that came back where we did the same assay. And what you will see is that after splashdown, I mean, you already see how chaotic that looks here. So there is nothing organized at all. Also, the cells, when you look at viability, they are alive. They're just really completely disturbed. But if you look like 2 weeks after the splashdown, and then 3 weeks and 4 weeks, you do see how they recover, and the closure of the wound gets faster over time. So those stem cells can recover here. Aging is reversible. So that was great. However, in the tissue chips, we had those cells together also with immune cells. And when we try to recover those, you see here that absolutely nothing recovered. They were just chaotic. They are viable. So if you do a viable stain, they are all alive, but they are just not functional anymore when they were exposed to those HD immune cells. And we could not reverse it. So the hypothesis was partially for the control group, it was proven, but not for our research group. So I wanted to use a few slides just showing you when we got into that mission in the beginning and really didn't know a lot of spaceflight. And Xiaomeng and Grigol are here today from the lab who did all the work. We were really excited about it, motivated, but our job was we were in UCSF and we have to get our chips ready and they need to be transferred to KSC for the launch. And as Charles already said, you have only one shot. So that was a Saturday morning, December 1st. And we are in the lab plating all the chips you see here. It was a lot of work. And you see here our picture. We looked happy. Everything looked fine. Unless at 9 a.m., we get the call from World Courier, who was supposed to pick them up at 1 p.m. to make that launch. They call and they say, we are not coming. But we needed the World Courier because they had the incubators with the temperature and the CO2 and the O2 to keep our cells alive in those chips. There was not enough time to abort, go to KSC and do it there. We would have needed to abort the whole mission. So you can see already the faces showing us also here in the audience. We were really disturbed. And that was 9 a.m. So we had a few hours to figure it out. And then we became creative. So at noon, wait a second, there is a, yeah, I think that's fine. I'm missing one slide here. I don't know where that went, but that is fine. I just explain it. So the first idea was, okay, we need temperature. That's really crucial for our cells to survive, 37 degrees. So we said, okay, reptiles, you ship all the time. They need to be at a certain temperature. We go to Petco and we buy those reptile patches where you can send the reptiles. It was Saturday morning, San Francisco, awful crowded. I went to Petco. I was the only one with a car on that day. So I went to Petco number 1. They were sold out. I went to Petco number 2, sold out. Petco number 3, no, we only have it online. I'm like, online is not helpful because the World Courier is coming. So then we said, okay, that is a disaster. And then I remembered when I have back pain, I put those patches on and they can hold the temperature quite well for 16 hours. And we only needed, I think 6 hours when I remember that one. And we measured that how to insulate that into our little package to keep the temperature we needed. And you see here how we packed it then. And we made it to that not World Courier, it was then I think FedEx or someone else. And they just took the box and transported it to KSC. Xiaomeng took the next flight to see in KSC if really everything is fine. And it was. They check here. That was Bill from Cases. They checked the cells and everything was fine and we could launch. But that was the first taste of what it means to do research in space. So I can only, when you talk it, I love the stories and we learned, we always now have backup plans. But that was one of the stories. So we are really excited about space travel, etc. But we are even more excited. What does it really mean for human health? And I told you a little bit what we are interested in. We're really happy that last point here is really, and it's supposed to be manufacturing. I apologize, stem cell manufacturing. We just got a NASA grant where we want to manufacture stem cells and use space conditions to have really perfect iPSCs, because iPSCs on ground, they tend to differentiate and don't stay in the pluripotent state. And that is really a problem for regenerative medicine. So we want to use the learnings we saw with the stem cells, how great the conditions are on the International Space Station, to get a little bit more information here, how to use really space conditions for manufacturing and therapies. And I have to thank so many people, but I think I focus because please, if you have questions about the studies, on Xiaomeng and Grigol, because they are really the drivers behind everything. And I want to mention our BioSurf implementation partner, because after the shipping disaster, we started working with Stephanie from BioSurf, and nothing like that happened again anymore. So they were really amazing in implementing everything, and it's just a pleasure to work with them. And then we have our collaborators mainly at MIT for those projects. Thank you.
Thank you, Sonia. So impressive. Just to right size your expectations, we do have a reception after from 8 to 9, but we still have an interesting discussion to be had here. So I'm going to ask our do-it-yourselfers to get your own chairs and set up your panel here. And while you're doing that, I'm going to make just a very brief comment. I didn't present my work tonight, but I have some of my interns here, and we do quite a bit of work in UC Space Health. I wanted to put the QR code up there. If you're interested, please log on. You'll see our projects. We do work in remote monitoring in Antarctica as an analog for space and as an analog for remote care. I'm working with a team here on a knowledge graph where we're working with gene labs and we're looking at multi-parametric prediction of different diseases. We're also working on a bed rest study with NASA over in Cologne, Germany, measuring bone loss with a very interesting point of care ultrasound device. So we have several different projects going and papers going all the time. So if any of this interests you, please let me know. And finally, I just wanted to mention, I was recently appointed to the National Academy of Sciences lunar expedition for humans to do exploration on the moon away from just the South Pole. So I did want to highlight my moon shirt. Okay, thank you. I'm going to hold one microphone back towards the end. We'll probably try to get a couple of questions in. I think we should switch to the next slide here. We tried to cleverly keep the light off our faces. How'd we do? Okay. So first of all, this is truly going to be an auto conversation. I'm going to prompt it a little bit, but as soon as they get near each other, the conversation just gets really vibrant and interesting. But I'm going to start, first of all, by saying, you noticed this was the Out of This World Science and Innovation Symposium. And for us at UCSF, if we have something that we identify as a novel, potentially impactful idea, we feel obligated to scale it up and out and go from idea to impact. And so I'm really interested if you could give us a few comments around how you think this incredibly challenging environment spurs on your innovation instincts and your innovation capacities. Anyone jump in?
Well, I'll start. That was a really hard spacewalk, by the way. Like there was a little PTSD when I saw that there, but that was the fun daylight part of the spacewalk. The dark part is much less fun. But the constraints that are placed on you in the space station, the fact that it's so closed loop, and the fact that you've just got all of these limitations on it, I think what it does is that you just deconstruct the way you think about doing an experiment. And I think all of these researchers are going to tell you, like, you start your experiment planning for space and it's like, whoa, this is not the way that I've done experiments before. And you have to think very carefully about, some really interesting things like a non-technician. You don't necessarily have an astronaut who's got a PhD in molecular biology is doing your experiments. Like the cold chain problems manifest and, the cold chain or the warm chain, like no matter how you're trying to get your samples to space. And so you start planning from the ground up for a constrained environment. And I think that is the thing that's actually going to drive that innovation. And that doesn't mean that you have to all send your experiments to space, right? But just think about trying to do your experiments in a really resource constrained environment, or your sample collection, or that kind of thing. And that'll change your thinking about the way that you think about your hardware development and you think about your consumables.
I think you learn also a lot about resilience. To not give up, and even if you think it's too late, you just push through. And we loved actually thinking about to simplify the experiments. Really, you can design it so complex in a normal lab setting, and if you're well resourced, you don't really think about it. To simplify, streamline. Maybe what we did also, we changed our media instead of every other day, and our team members had to come in on weekends. We simplified it and made it easier, and that really forces you to do that, unless if you're like in a well-funded lab, you might not think about it and you just go with it because everyone did it like that in the past. So I think for us, we took that as an opportunity.
I actually view like research in space as very similar. It's very structured. So I view it in some ways very similar to kind of a regulatory environment where the procedures that are done, for instance, in the clinical lab, there's a ton of like QC and QA, basically quality assurance and quality control. And just to make sure that, because you can't afford, you can't afford a test to fail, essentially, because patient care is at risk. And so I view that I think it's really helped to be able to structure an experiment in space, because you really have to think about every detail, and areas that your experiment could go wrong. You have much less flexibility, but at the same time, it also gets you better organized. And we actually used to joke, but the fact is that the data that Kate generated in space was actually superior to the data we generate on the ground. To give you an example of how working in such a structured environment can have several positive aspects.
One of the follow-on technologies that we're thinking about is if we're thinking about biomanufacturing in space, how to get these cells back to Earth. Right? If we were thinking about using it and giving it to patients, they have to be alive and functional. So how to do that transport? So one of the things we're thinking about is advanced approaches or technologies for cryopreservation and code preservation. And so we do have a project that is going to test that during launch forces. But that technology, even if we didn't go to space, would be highly impactful for tissue engineering, cell therapy, and transplantation on Earth. So I think a lot of these sort of necessities that we think about for what we need to do to get experiments or things done in space have high impact for Earth applications on its own.
Thank you very much. I also heard you say things about having to be persevering. Let's talk a little bit about funding and how hard is it for you to convince funders? Okay, present day accepted, but so how hard is it to convince people that this work is important and why, and what sort of strategies around that? And then the second part of that question, pick either one, is what are some novel avenues for funding that you hadn't really been exposed to before that have opened up because you're doing space research?
For us, it was really, I think, reaching the high school students. So we worked together with Orion Quest. So they made our project for the schools, and we go to the classroom, they can analyze the samples. So usually, at least we didn't have that exposure. And really then to motivate the next generation of scientists is really, we think, is one of the biggest pleasures we have due to the space research, I would say.
Yeah, I think so. Right now, ISS is the only place that you can send your experiments to, and it's really complicated. I work for the government, and it's really hard when you work with the government, right? There's a lot. It's not customer friendly. You guys would agree. There are commercial stations that are coming online, and for them to survive and thrive, their business model, they're going to have to be customer friendly. And so I think if these stations do come online and they're successful, the path to space is going to be a lot easier. That being said, NASA's funding has gotten cut like everybody else's. And if you're going to go on these commercial stations, it's pay to play. And so I think you then need to bring a third party in that's going to be your funding source, and good luck fishing in the NIH and NSF pool these days. So I think we're going to have to get really creative. I think a lot of these things appeal to philanthropic donations. We're talking about things like aging and cancer and stem cells. Like this is an immediate impact on human health. And so I think getting this message out there to donors is important. It also, people love space, right? It can crack that door for you when you're talking to a donor, or philanthropy. I think when we're also talking about commercial space stations, we can talk about production of things. And so in that case, maybe companies are interested, maybe venture capital is interested, and I wish we had all the funding in the world to do the basic research, because I think that's the most important thing. But if somebody says, hey, we think we've got a commercial angle on this, and we can use that funding to do the initial development. And at the end of the day, we want to put this into patients, and we're not going to do that, right? As just research institutions, having the scale and the manufacturability. And so if there are people that are interested in manufacturing in space or using space to understand your manufacturing R&D process, so maybe not actually doing it in space, but things like Keytruda, where you're understanding that process and you can bring that back and you can replicate that on Earth. I think commercial sources of funding partnered with academia and government might be a really good place to go.
Oh, yeah, maybe just to add on what Kate said, I really feel that with the emergence of kind of commercial interest in space, that there will be opportunities. For instance, I know that, SpaceX, for instance, as a mechanism by which you can just submit your idea or proposal, and if it's if they deem it interesting or worthwhile, that's something that they'll consider. There are opportunities such as kind of the Humans in Space initiative, which is launched again for this year as well. So if you want more information about that, you can talk to me about that, and I can. And they're planning on funding more projects this year. So it's increasing in size. So I think with the availability of commercial funding and partnering with the government for getting your experiment aboard the space station is certainly a feasible route.
Great. I really want to thank you for this. I'm going to ask one other question. When I think about commercial spaceflight, one thing I think about, because part of my work is in monitoring, trying to understand what's happening to humans in the real world environment, like away from the clinic. So that capability is really important. And we have a highly selected, highly trained astronauts that we're sending now. But as you see on the commercial space flights, the criteria are much different. Like, do you give a good interview? How big a check can you write? So the substrate is going to be very, very different and much more diverse. So we do actually have an imperative now. And it's another forcing function for doing a better job of monitoring and evaluating people on Earth continuously. I'm going to tell a quick little story because I think it's true, but it could be a lie, but she told me I could tell one lie. But I remember when I was asked to be the Chief Health Innovation Officer for this innovation arm of NASA called Translational Institute for Research Space Health. And that's actually a great source for people who are interested in funding, but also for training opportunities. I was really surprised because I had been covering ocean rowers and extreme athletes and remote athletes. I was very surprised that the least monitored part of the entire spacecraft system really was the human. And it was really stunning to me because as sophisticated as it was, the craft was incredibly monitored and lots of closed feedback loops, et cetera. The human was sort of spot checked. And I'll let Kate speak to this, but she also apparently had her own frustration with that. So she decided, I am monitoring myself here. Why am I not doing this up there? And so tell them what happened when you decided you wanted to take your own watch up.
Oh, yeah, that was really fun. So, this active watch thing, I wanted to take a look at my sleep and wakefulness patterns, and you just couldn't get it through the 8. They said, well, first you have to buy 10 of them and go to White Sands and blow them up and make sure that they're not going to blow up spontaneously. I'm like, look, if the Apple Watch was blowing up spontaneously, like, we'd see some headlines about this, right? Like it's going to be fine. But this is sort of, how NASA works. And it finally took the SpaceX crew who wanted to wear their watches in the SpaceX capsule. And then SpaceX went to NASA and said, we need to approve these watches so that Crew 1 could get their watches on Earth up to space. And so I think there's some driving function that we can work on with like consumer technology and sort of this forward-facing thing. But I think you've got a really great point about the range of health issues that we're going to see. So astronauts like our physical is the scariest time in the year for us because if they find something, we're disqualified from spaceflight. Like we screen people incredibly harshly, and we disqualify for things that could be a mission risk. When we're talking about commercial spaceflight, and this isn't just billionaire tourists. When I say commercial spaceflight, a lot of this is also other government agencies, countries that have not had a spaceflight program. The net is cast much broader, and so we may have we're still going to screen them, right? If there's an overt health problem that could be dangerous to them, we're not going to allow them to go to space, but we're going to be much more lenient in terms of what they're taking that risk, right? That, something's going to happen. They can't complete the mission. They're also going for shorter periods of time. They're not really going for more than a week or 2 weeks, whereas the astronauts have to be up there for 6 months up to a year. And so health conditions could come on much faster. And so what we're going to see when we start sending people with a range of health conditions is what happens to the human body that's not, a military aged male or female that's been extensively trained and has, works out, 2 hours a day as mandated by NASA. What are we going to see that's like subclinical. I think the continuous monitoring of the human is going to be super fruitful when we're talking about things that maybe have, people have different states of health and disease, and what are the spaceflight impacts that we are not seeing because we're selecting for healthy individuals.
And that leads me perfectly, thank you, into the next question, which is really understanding more, which you can see the drive on our campus too, of understanding the context, the human health in the context of the environment and the health of the environment. And I think, Charles, we had an interesting discussion today, and I think each of you consider this as how much is it not just the pathogen, but the host? What influences how this is going to express? Would you like to, any of you can speak to this?
What's interesting is I sort of began my career as a microbiologist, infectious diseases, but now I'm sort of moving actually away from the pathogen, and my lab is now actually focused on the human host, on the patient, because my feeling is ultimately disease, even in infectious disease, it involves an interplay between the pathogen and the host, and clearly the host response plays a very important role with respect to the disease. I mean, you can have a single pathogen like West Nile virus. 80% of patients with West Nile virus have no symptoms at all, 20% have symptoms. Only 1% have like fatal encephalitis or potentially fatal encephalitis. And so all of that is really based on the host response or the genetics. And so my feeling is that if we can develop agnostic technologies that will allow us to, say, do continuous monitoring for not only for infections, but really for any kind of acute or chronic illness, things like sequencing, proteomics, looking at, cytokine profiling or immune cell profiling. I mean, these are agnostic technologies that can apply across a wide variety of fields. And it's going to be really important if we think about bringing a laboratory in space. We're never going to have, I think it's going to be a long time before we'll have like a fully functional lab up on the moon or on Mars. So the idea is that we're going to be forced to develop individual assays that broadly can survey human health overall. And so facilitating those technologies, and I feel that the technology has advanced to a stage where we could feasibly develop, for instance, an omics-based assay that can do that for us, that can diagnose a range of different infections and non-infectious conditions.
Thank you. And I'm going to let you pass the mic along, and any tips? How many people in the audience would consider themselves learners? Learners. I think it's everyone, right? But in terms of our academic hierarchy, because I know we had quite a few faculty sign up, but we also had a tremendous number of students sign up and postdocs. And the reason I'm asking that is because I think that there might be a tip from each of them, and you can ask them individually as well. What would you say in terms of how you might just first dip your toe into this field?
I mean, if you have interest, what I did from the beginning, I would always like volunteer or do shadowing, also really where I'm already interested, and I wouldn't go in any other areas. And that then gives you the contacts, and you go a second year and you really grow into it. And it also helped me when I started. I'm a trained cardiac surgeon. I thought that's what I love. And I didn't. So I also, the path is not straight, but I think you have to understand what you are really motivated about and love, and what passion you have, and to follow that. And I think I would just follow the passion and see then where the path goes.
I would just second what Sonia said. I think that's just so cogent in terms of both just going out and seeing, knock on doors and trying things, and then finding your passion. I think that's absolutely the key.
So I have two things. The first is just like personal advice, where you are in your journey and your career journey, is that scientists love to talk about their work. It makes them so happy to talk about their work. And so the knocking on doors thing, I mean, I got into Pat Brown's lab because I emailed him and I said, I think microarrays are the coolest thing. I'm willing to come up and start grad school early so that I can work in your lab, because it was really hard to get a rotation in the lab. I've read the last 5 of your papers, and like, can we meet? Like, I was, like, gosh, how obnoxious was I as an undergrad being like, I must work with you. But, Pat he was an amazing mentor. But, reading about what people are doing and just asking them to talk about it. People are so excited to talk about their science, and that can open a lot of doors. So don't, come in knowledgeable, make sure you read at least like their last 2 papers. But, you know, scientists just love that. It makes their day. It's going to make them happy, and then they're going to want to hire you. So definitely do that. I think also be cross-disciplinary, be, you know, multidisciplinary, take whatever you're doing and then try to find an opportunity to do something completely different. And whether that's from grad school to your postdoc, or as you're doing rotations, you know, your clinical fellowships and what you're interested in clinically. You know, I'm super cross-disciplinary, absolutely by accident, because I applied to be an astronaut while procrastinating writing a grant application at MIT. This is a true story, not a lie. You know, and I've just kind of gone into these different, really radically different institutions. NASA is an engineering organization. I'm not an engineer by training, and like lots of culture shock the first few years there. But getting into something that's a completely different field, that is a different way of doing things, then you can be that cool intersection person that's like, you know, I combine this, and you're multi-hyphenate, and then everybody really does want to hire you. So that's my best advice.
Did you want to make any other comments, Joyce?
Oh, yeah, I'd like to second what Kate said about being interdisciplinary. For instance, I began as electrical engineering. I went to computer science. My classmates went on to Google and Yahoo and other startups at the time, but I ended up going to medical school, and then I did biophysics, and then I went to genomics. And don't be afraid of new experiences, and especially now with the advent of, you know, AI, for instance, and programming. So having, say, a biostatistical, I think, a bioinformatics background or programming background is going to be very, very useful, I think. So especially if you're interested in learning new things. And science nowadays is very highly collaborative as well as interdisciplinary, and being able to, you may not need to master these individual fields, but to be able to communicate with others in the fields. I mean, you may not need to be a bioinformaticist, but you probably will be hiring bioinformaticists. So being able to kind of talk to them in their language is going to be really important.
I think that's a great place to wrap it up. And I think the point is that healthcare, research, and innovation, they're team sports. And that's why this is a real honor for me to consider this group of people colleagues. It's an incredible honor that we can be face to face with Kate. That's what this picture represents to me, no matter where in the galaxy she is. And then also to hear our own colleagues talk, each of them speak around the world, but we almost never get to hear each other and be in conversation. Take advantage of this. This is a rare evening. And your patience has been extraordinary. So you'll be rewarded with food and beverage, but we'll all be around for a while. And I owe a lot of thanks. This was co-hosted by the Center of the Innovation Ventures Group, but also by Department of Orthopedic Surgery, UC Space Health. And I see most of the people that help volunteer, clinical research coordinators, our own assistant vice chancellor from Innovation Ventures. We had a lot of people helping. I think they're already out there waiting to serve you, and our great caterer, Dee Wagner. So thank you. And thank you to my extraordinary colleagues. And thank you for showing and staying. And we'll just start this conversation and keep it going. Please connect with us.
Nice meeting you. Nice meeting you too. Yeah, fantastic.